Thank you so much, Mariana. Super kind introduction. Just a second, let me try to... Can you see the slides without the notes?
Yes.
Okay. Thank you so much. It's really a big pleasure for me as well to be here, and thank you for the invitation. And I think I have to congratulate you as well, because it's a super timely initiative, I think, to have a startup like focusing on disseminating like scientific knowledge, and especially in a time that we have so much spread of misinformation. And it's great to have you guys working towards this goal of disseminating knowledge. And I'm super curious to see how it's going to turn out. Good luck. And yeah, for me, it's a great pleasure to talk about also one of the most fascinating topics for me in biology, which is how like DNA orchestrates the dance of life. And I would like to start by calling your attention to a process that is really close to my heart. I've been working on it for a few years now. It's embryonic development. So this is about like how super creature, like in this case a turtle, that has lots of specialized structures like limbs that allow for locomotion, eyes that allow for color vision, and in that case a shell that allows for protection against predators. How such creature can develop from such a simple start as a single cell, So what I wanted to highlight here is that what is in the core of this process, it's the molecule known as DNA. So it carries the instructions for a single cell to make this super complex organism. So DNA is really the basis for heredity. So heredity, simply put, it's a process by which the children look like their parents. So it all starts when a sperm cell carrying the father DNA encounters the egg cell, which has the mother's DNA, and then the fusion of the cells kickstarts the process of development. But how does information flow then from the DNA to drive this whole process? So just as a recap of what DNA is, so it's really, you can think of it as a Lego, right? So it's a chain of building blocks, like essential components, the A, C, T, and G that Mariana just talked about. So it's a molecule that has all these building blocks sequentially, and they can pair with complementary letters. So in the case of the A, always pairs with the T. In the case of the C, always pairs with the G. So then it forms this long chain, and it is located in the nucleus of the cell. And you can actually, by doing experiments in the lab, you can observe the DNA under the microscope. So we can use a chemical that makes the DNA shine under the microscope. And this is represented here, where you have two cells where DNA is the blue, in the blue area here. Also, DNA, it's not just randomly organized in the nucleus. It actually forms very complex structures. So each DNA molecule is organized in the chromosome. So here we can have, you can see the two different chromosomes labeled inside the nucleus. So they occupy very distinct spaces in the nucleus. So the central dogma of molecular biology is being known since decades. So that's the most basic way to explain how information flows from the DNA. So essentially, the DNA molecule serves as a template for RNA molecules to be produced to the process of transcription. So once you have mRNAs, this can be translated into proteins. So yeah, this is the central dogma, essentially. And these processes, they follow very precise rules. So the DNA, like there is always a correspondence between one letter to one letter of the RNA being produced. So you always produce this new strands of DNA of RNA based on the template here. And next, when the RNA is being translated into the protein, then it works as trios of nucleotides, of letters. They will give rise to single amino acids, which will form protein as well, just as a Lego like the DNA. And the proteins that are formed in the end of the process, they will be the responsible ones for executing the chemical reactions that happen inside every cell. So all these processes, they happen inside cells, as I just told you, and the cells themselves, they then they organize themselves in complex manners, forming tissues, which will have a function like supporting organism to breathe or to see colors. And the tissues then organize into systems like the respiratory system or the nervous system, and the collection of systems forms the organism. But today, really, I wanted to call your attention for the DNA. So we've learned a lot about the DNA in the last century, actually in the 20th century, but I wanted to focus on one milestone, which is the Genome Project in the end of the 90s. So that's when really we have, for the first time, all the DNA sequences for an individual. So we had genome projects for human genome, for fly genome, for mouse, and other species. So that was the first time we had the genome sequence. And it's allowed us to see, like we already had evidence for this, but it allowed us to see in the whole DNA, like which sequences can be transcribed into RNA. So these are the so-called genes, right? And this one example of how a gene looks like in the human genome. So it's the gene called Kytos. It has 6,700 letters. And it's it's a small one for the human standards. But what was really interesting, in my opinion, from this genome project is that we could see that the majority of the DNA is actually not transcribed. So it's only, even though we have more than 20,000 of these portions that are transcribed, the so-called genes, it's less than 3% of the whole DNA that is present in the individual. So now I wanted to introduce you to a key concept that's probably one of the main messages of the talk. It's like how then you can create diversity from such a system. So if you think of the DNA containing many genes, like in a complex organism, we cannot have all the cells having all the genes active, transcribing all the time. Because otherwise you would just have like cells that are just similar to each other, right? So you need to have a situation like this where you have some genes being switched on, some genes being switched off. So you can go from a situation that you have like very, very homogeneous, simple few cells into a very complex structure, like an embryo. So in other words, you need to have like one cell in which certain genes are active, in other cells that maybe the same gene A here is active, but then the gene B is not, instead of the gene F, and so on. So every cell has a different combination of genes being active and inactive. And so this will lead to different structures being formed. So how does this dynamics then come about? Like the genes themselves, they are not responsible for their own transcription. So they need other sequences that help them to be transcribed. So I wanted to introduce the first one here is the promoter element, promoters. So every gene has a promoter, it's a relatively short sequence that is next to it, and it controls the activation of the gene. So essentially what it does is to recruit a protein called RNA polymerase, which will open the DNA strand and will use one of the strands as the template to generate a new RNA molecule. So I told you before that the information flows from DNA to RNA to protein, but actually you also have like proteins coming back to modulate the system. So it's not a direct conversion back to RNA into DNA, but rather the proteins are acting on DNA to stimulate the information to flow on the right direction. But then promoters, they are simply the initiator of transcription. They cannot determine if the gene is going to be on or off on that cell. For that, we need other type of elements called enhancers. So these elements, they are shown here in green in this cartoon, and they are responsible for regulating when and where certain genes are activated. So they can act in different ways. They can activate genes that are very close to them, but they can also act from long distances. And sometimes multiple enhancers can activate one gene. So it's a very, very complex relationship, really. So enhancers, they need to come in close contact with the promoters to activate that gene. And just to show you like examples of how enhancers can be specific to certain cells, here I took images from an experiment that we can label in dark blue the cells where a given enhancer is active. And here you have two mouse embryos, and the one in the left, you have an enhancer that is active in the head of the mouse embryo. And then on the right, it's an enhancer active on the limb region. It's very specific. So, but that's actually not the whole picture. There are other elements that have been also discovered that have other modulation roles. So, for example, silencers, they can have the opposite role as enhancers. So they rather shut down a gene in a certain cell. Or insulators, they can have more complex specific roles. So they can either activate or inactivate a gene depending on many factors. But that's the point here is not to go in detail on how these things work, but it's just to give you a picture of how complex actually gene regulation has to be in order for complex creatures to be developed, right? Okay, so then we went from this picture where we have like certain genes in the chromosome, in the DNA, to now a picture that we have a lot of other elements there that regulate when and where these genes are active. So just to give you overview of how diverse these enhancers can be. So it's a panel of lots of fruit fly embryos here. So each of these American football things here, they are a single fruit fly embryo. And in dark, it's where a single enhancer is active. So you can really see that there is like a lot of nice and beautiful patterns. But in some cases, it's actually very simple. You just have it in a few cells. And that's the same for mouse embryos. So this is a feature that is present in like all animal species, really. But so far I've been describing to you like how the gene regulation system works, let's say in a healthy case. But in biology, we are also interested in what happens if we disrupt some of these elements. Like in the case that we have a mutation in one of these elements, we have the letters being deleted or changed by a different letter. So I'll show you one hypothetical example here. So let's say we have one enhancer that has this sequence, And this enhancer is driving expression of a gene in a hand. And it's important that this enhancer functions so we have like a proper hand being formed, right? But if you have a mutation in that sequence, like an addition of these red letters, we might end up in a situation that we have alteration in the formation of the hand, and suddenly we have very dysmorphic hand in comparison to a healthy individual. And because this enhancer is important for regulation of the genes in the hand, then you wouldn't have other effects in the body. Like you can see that the ear and the arm, they are normal, but it's just really that specific tissue that is affected. So mutations in general, they can cause disease when we consider them in one or few generations. But it's the same mutations, they will be important across thousands to millions of years to generate the diversity of organisms that we see during evolution. So all the types of shapes, of body shapes, of sizes, of colors, are really like a result of how the DNA has been changing between species across millions of years. And we know now that the evolution of new traits, it actually, very often it's related not to the appearance or disappearance of genes, but rather to the reshuffling of the enhancers and which are their targets. So we can have the situation that one species has this configuration here, but then it loses certain enhancers, and some enhancers, maybe they were not active before, but then they become active now, and this will drive the appearance of new traits. You can think of all ways that this can be reorganized. So I want to show you, I would say, I guess, a more visual example of how this can be. So here we have a very complex DNA sequence, and we have one enhancer that activates one limb gene very far away. So then if we take the sequence of this enhancer, we can compare it in the mouse, in the human, and in the snake. So in mouse and human, it's both species that have limbs. So their sequence is actually like very, very similar. It's almost identical. And then in the snake, which is an animal that doesn't have limbs, then we lose the red region that I highlighted here. So to know if this change is actually involved in the loss of limbs during evolution, what some researchers have done is to first just look at the mouse development, like normal mouse. So it has forelimb like this, hind limb like this. And then what they did was to replace this enhancer by a human equivalent. And then they saw that, the mouse, it's pretty much normal. So it has a forelimb and hind limb, just like the mouse with the mouse DNA. But when they put the snake DNA, they replace the mouse sequence by the snake sequence, then you have really a lot of problems in forming the limbs. So then you have like a near absence of the limbs in that case. So I think this is an interesting example of how evolution can act more in the regulatory DNA, in the enhancer DNA, than in the genes themselves, which will form the proteins. The thing is that we're still quite far from really deciphering the instructional manual that is the DNA, right? So just to give you an example, let's take this very complex region where you have a gene involved in the limb formation, one gene involved in the brain formation, one gene involved in the heart. And you take the sequence of these non-coding regions here, and then you make mutations in all of them. it's very difficult to predict what's going to be the outcome. Like, will the link gene be, we will have like problem with the link gene, and then you have link defects, or is it going to be more the heart, or all of the systems will be affected? It's still difficult to understand. So there are some new technologies that are playing a major role in advancing our understanding. I want to highlight two here. So it's technologies to read and to write DNA. So the first one is the next generation DNA sequencing. It's been evolving a lot in the last 15 to 20 years. And it's now it allows us to sequence DNA from thousands of individuals and thousands of different species. And it's becoming also much more portable. So people can actually take these sequencing devices to nature and sequence, for example, DNA from endangered species. So it's really like spreading a lot, this type of technology now. And the other one is our ability to rewrite DNA with, for example, CRISPR. You might have seen that in the news. So the researchers that developed this system won the Nobel Prize in Chemistry in 2020. And it basically allows us to do precise editing of genomes. So you can take a given sequence like this, and maybe you can, you want to insert a chunk of DNA, or you want to delete a chunk of the DNA, or even make a very fine manipulation, like just changing one single letter. And this is possible now with this type of technology. So this, so we, as a community, I think biologists are going towards understanding the how this instruction code works, and it's mainly a work in progress now. So I'm actually reaching the end of the presentation. Was faster than when I was trying it out. Okay, but what I wanted to leave as a take-home message for you is that just as an overview, like information flows from DNA into to allow development and evolution of organisms. So we have all these transcription, translation processes happening in the cells, and the initial transcription is controlled by a very complex combination of elements in the DNA that we are starting to understand now. And this really allows like a single cell to develop into an organism. If this goes wrong, then it can generate disease, but it can also allow evolution to happen and new traits and adaptations to happen. And that's it. Thank you all for your attention, and I'm happy to discuss more. If something is not clear, you can ask me, and I can explain again. Or, yeah, I'm happy to talk.